Hi, welcome to my presentation on Chapter 5 on the carbohydrates. So carbohydrates are the third type of biological macromolecule that we'll go over. We already talked about proteins and nucleic acids, so now we'll do carbohydrates, and then in the next chapter we'll cover the lipids. A carbohydrate is basically just a sugar, or sometimes a larger molecule that is composed of sugars. The main function of carbohydrates in cells is to store energy, but they also have some other functions. So for instance, you need them to build your nucleotides for nucleic acids. So every nucleotide has one part of it that's a sugar, either ribose for RNA or deoxyribose for DNA. So if you don't have those sugars, then of course you can't build a nucleotide or the nucleic acid itself. So that's another function, and then also they have some structural functions as well. So sometimes a carbohydrate is used to build a structure in an organism. So for instance, cellulose you've probably heard of in connection with plants. Cellulose is a major structural carbohydrate that's found in plants. So we're going to go over the functions of carbohydrates in more detail kind of at the end of this presentation, and in the first part we'll focus more on the structure of carbohydrates. And we'll start off with some kind of vocab words related to carbohydrates. So first, a saccharide is just a sugar. So anytime you see the word saccharide, you know that whatever larger word it's a part of, that means that there's a sugar somewhere in there, and it's a carbohydrate. Then you have a monosaccharide, which is just one sugar. So like glucose is a monosaccharide, and fructose is also a monosaccharide, just single sugars. But you can also link two sugars together, and that gives you a disaccharide. So sucrose is actually a glucose molecule bound to a fructose molecule, and that gives you the disaccharide sucrose. If you keep on adding sugars to your disaccharide, once you have a few of them on there, you're going to get an oligosaccharide. Oligo just means a few. So an oligosaccharide is a few sugars that are linked together, usually to form a chain. And then finally, the biggest carbohydrates are polysaccharides. Poly means many. So a polysaccharide is many sugars that are linked together to form a chain, and sometimes you might have branches in that chain as well. So today we'll talk about kind of the structure of monosaccharides, and then a bit about disaccharides, and then we'll finish talking about polysaccharide structure, and then we'll move into the functions of carbohydrates. Just like for enzymes, you can always recognize enzymes by the name because it always ends in ase. It's the same thing for carbohydrates. You can always recognize a carbohydrate because its name is going to end in ose. Everything whose name ends in ose is a sugar of some type. So ribose, glucose, sucrose, fructose, lactose, cellulose, these are all different types of sugars. Cellulose being actually a polysaccharide, so many different sugars linked together in a chain. But since it's just made of sugars, it's still going to have an ose ending. So first we'll go over the structure of a monosaccharide, or a single sugar. Is basically a skeleton of carbon atoms with different things stuck onto the carbon atoms. You have to have at least 3 carbons in a chain to get a sugar, but you could have more. Most sugars that we think about have 5 or 6 carbons, but there are sugars that have more like 9 or 10. They're just not very common. If it has 3 sugars, then if a sugar has 3 carbons, then we call it a triose. Any sugar that has 5 carbons is called a pentose. Any sugar that has 6 carbons is called a hexose. So actually every sugar can be kind of classified based on how many carbons it has. And there's kind of general names for sugars that have certain numbers of carbons. Not that you need to know all of those names. On each carbon atom in that chain, there's 3 different options for what you could have attached to it. Of course, it could be attached to other carbons in the chain, right? But apart from that, it could be attached to hydrogen atoms. It could be attached to hydroxyl groups, which is just an OH, or it could be double bonded to another oxygen. If you have a carbon that's double bonded to an oxygen, that's a carbonyl group. Most sugars are going to start as a line. Well, I guess I should say sugars in general start as a line. When you put them in water, a lot of them will actually form a ring, but if you have them just as a powdered form, they're going to be like linear, so just the carbons in a chain. In one of those sugars that's like linear, you're always going to have one carbonyl group. So the other carbons that don't have the carbonyl group, they're only going to have hydroxyls attached to them or hydrogens attached to them. No more carbonyls than just the one. So you've got a couple examples of sugar molecules over here. These are really simple sugars. You can see they only have 3 carbons. So these are both trioses, and they're the simplest type of sugar that you can have. If you have one less carbon than this, then it's not a sugar anymore. You can see that each of these has one carbonyl group, one carbon that's double bonded to an oxygen. The carbonyl groups are located on different carbons for these different sugars. And the other carbons are going to have hydrogens attached to them and hydroxyls attached to them. So actually each of these other carbons has just one hydroxyl group attached to it, and then the other bonds that it can make are just filled up with hydrogens. The same thing for this carbonyl. The other bond that that carbon can make is just occupied by a hydrogen. We always number the carbons in our sugars. So similar to how in a nucleotide the carbons in the ribose or the deoxyribose are numbered, the carbons in any sugar would actually be numbered. And just by convention, we're always going to start numbering, of course, at the end, at either end of the chain, but we're going to start at the end that has the carbonyl group. So in this case, this carbon at the end has the carbonyl group on it. So that is the carbon we'll start numbering at. So that's going to be C1, or carbon number 1. Then we have C2 and C3. With this other sugar, the carbonyl group is actually in the middle. For this one, it doesn't really matter which side we start numbering at, since they're both identical, but we'll just start on the top one. So that's carbon 1, carbon 2, carbon 3. If this was a 4-carbon sugar, so if you had another carbon coming off down here, then the top carbon on the end would be closer to the carbonyl group than the bottom carbon, and so we would still start numbering at this top carbon because it's closer to the carbonyl. So I kind of mentioned that we have names for sugars that have a certain number of carbons, like any sugar that has 3 carbons as a triose and any sugar that has 5 carbons as a pentose. We also have other names for general types of sugars. So any sugar that has its carbonyl group at the end of it is going to be an aldose. So on the top here we have an aldose. The carbonyl group is located at the end, one of the end carbons. But you might have it somewhere else. You might have it in the middle or just on any other carbon that's not at the end. In that case, we would call that a ketose sugar. So a ketose, you know it's a sugar because it has the ose on the end. Same for the aldose. Since it has the ose, you know it's a sugar. But with the aldose, the carbonyl group carbon is going to be at the end. And with the ketose sugar, the carbonyl group carbon is going to be just somewhere that's not at the end. So that means we've actually kind of gone over 2 of the ways that monosaccharides are classified. They can be classified by how many carbons they have, as trioses or pentoses or hexoses. They can also be classified by where their carbonyl group is. So if it's at the end, it's an aldose. If it's not at the end, then it's a ketose. And then there's also one more way that you can classify monosaccharides, which is based on where their hydroxyl groups are pointing. So you can kind of see that with these ball and stick models over here. You have 2 different sugars here, glucose and galactose. These are both hexoses because they have 6 different carbons. And they're also both aldoses because their carbonyl groups are at the end. So then also that carbon that's at the end that has the carbonyl group, that's going to be your first carbon, and then you number from there. So based just on that, we would say that these are the same molecule, but they're actually not the same. They have a slight difference, which is just the way that one of these hydroxyl groups is pointing. So in carbon 4, in both of these sugars, the carbon is attached to a hydrogen atom and to a hydroxyl group, but it's actually not pointing the same way. So if you look at the glucose, the hydroxyl group is actually pointing kind of backwards. so that the carbon and the hydrogen atom are kind of on top of it, or they would be coming out of the page, and that hydroxyl group is going back into the page, or into the screen, I should say. But if you look at galactose, it's the other way around. So in that case, the hydroxyl group is the one that's kind of on top or pointing out at us, and the hydrogen atom is the one that's behind or pointing back into the screen. So that means that at that location, at the fourth carbon, they have a difference in where their hydroxyl groups are pointing, which makes them different sugars. So monosaccharides are always going to be classified based on how many carbons they have, where their carbonyl group is at in the sugar, and for each of their hydroxyls, which way is that hydroxyl group kind of pointing? Is it coming out at us, or is it going back into the screen? So the fact that you can have two of these molecules that are basically identical, like glucose and like galactose, but the only difference between them is the way that one of those hydroxyl groups is pointing, that's actually a concept called chirality. Chirality is kind of handedness in a molecule. So you've got your two hands, and they basically look the same, but actually one of them is a left hand and one of them is a right hand. So they're not actually identical hands. Like if you had two left hands, then one of your hands would have the thumb coming off on the other side, on the side that you're not expecting it to be coming off of. And if you actually take your hands and just kind of stack them on top of each other, then you can see that they're not identical. The thumb is coming off a different side of each of your hands. On the left side, or on your left hand, the thumb is coming off towards the left, and on your right hand, the thumb comes off toward the right. So even though they look basically the same, they're actually different. You can have molecules that are basically like that as well, where they look the same, but when you try to line them up, you see that they actually don't line up the exact same way, because the way that different things are attached to them is different. So in this diagram here, you have an example of a couple of molecules that have handedness or have chirality. So molecule A has a carbon, it's attached to an oxygen, or I guess I should say it's just, it's attached to four atoms of different elements. And on the other side, you have molecule B, which also has a carbon in the middle that's attached to those same four different elements. But just the way that those attachments is arranged is different. So when you look at these two molecules, they look the same, but if you were to try to line them up directly on top of each other, like first you would maybe flip molecule A so that the red atom is pointing out the same direction as in molecule B. So then you would get it looking like this. And then if you took molecule B and A and literally put them on top of each other, you would see that they're actually not the same. The red atoms line up and the black atoms line up, but the blue and the green, they don't line up. They're in different spots. So these are actually not identical molecules. One of them is kind of a left-handed molecule and the other one is kind of a right-handed molecule. And you can also see that up here with the amino acids. So these are both basic representations of an amino acid. In the middle you have your alpha carbon. Coming off of it you have your carboxyl group and your amino group and a hydrogen atom and the side chain of that amino acid. So every amino acid can have either a left-handed version of it or a right-handed version of it. And that just depends on the way that these other groups are arranged around that central carbon. So if you change the way that they're arranged, you're going to get either the left-handed version or the right-handed version of that molecule. Chirality can occur in any molecule that has a carbon atom with four different things attached to it. So it could either be four different atoms, like in molecule A and B here, or it could be four different kind of groups of atoms. So in our amino acids, this central carbon is always going to have four different things attached to it. It has a hydrogen atom, and then it has groups of atoms. It has a carboxyl group, an amino group, and a side chain. So those are all different things. Anytime a carbon atom has four different things attached to it, it is going to be chiral. It's going to be possible for there to be a left-handed version of it and a right-handed version of it, depending on how those things are arranged. If a carbon atom has like only three different things attached to it, then it's not going to be chiral. It has to have all four things that are attached to it being different from each other to be chiral. So for instance, with this amino acid, if it had 2 carboxyl groups instead of a carboxyl and an amino, then that would only be 3 different things. So it would be a carboxyl, a side chain, and a hydrogen atom. That would not be chiral. It has to have 4 different things attached to be chiral. The reason this matters is because sometimes in the body, you have like maybe an enzyme that needs to like perform some type of reaction on that chiral molecule, and it's only going to be able to recognize either the left-handed version or the right-handed version. So that means, for instance, for amino acids, the ribosome has to be able to stick them together and catalyze the formation of peptide bonds between them so that it can build a protein. The ribosome is only able to do that with amino acids that have the right-handedness. So let's say they have to be left-handed, and the ribosome can, it can combine amino acids that are all left-handed and catalyze peptide bond formation between them. But if you stick in a right-handed amino acid, the ribosome can't recognize it because it's not going to fit the active site. And that means that the ribosome will not be able to catalyze a peptide bond between that right-handed amino acid and any other amino acid. So for any molecule that has chirality, usually you have to have the correct version of it in order for it to actually function correctly. So if you need the left-handed version, you have to have that left-handed version. If you have the right-handed version instead, it's not going to work. It's not going to have the right function. So there are some molecules that doesn't really apply to, where the body can use the left-handed version or the right-handed version kind of just as easily. But for a lot of molecules, it has to be the right one. And that includes for sugars. So sugars are highly chiral. Most of their carbons are chiral carbons. Most of them will have 4 different things attached to them. And so that means you have a lot of options for kind of different places where the sugar could be left-handed or right-handed. And that is actually going to matter for the body. A lot of the time you'll need the left-handed version of the sugar, but you can't use the right-handed one. So maybe you need the glucose, but you can't use the galactose instead. And I guess before we move on, I'll just mention that this affects certain drugs as well. Like some pharmaceuticals, do have a carbon somewhere in there with 4 different things attached to it. It's actually, that's pretty common to find. So for a lot of those drugs, when you're producing it, you have to make sure that you're getting the right version. You're getting either only the left-handed version or only the right-handed version. For some drugs, the left-handed and the right-handed versions will work just as well, but for others, like only one version works and the other one doesn't work. And what's even worse, for some drugs, like one version works, like the left-handed version will work, but then the right-handed version is toxic. So you have to make really careful. You have to be really sure that right-handed toxic version is not in the actual product that you're producing and giving to people as a medication. So I mentioned that when you put sugars in water, or when you put monosaccharides in water, they tend to form a ring. So if you have the powdered version of that sugar, it's going to be linear, but if you put it in water, it makes a ring. And we're actually usually used to looking at sugar structures that are kind of in the ring form. And that's just because in biology, we're always thinking about organisms, and organisms are full of water. So if you have a sugar in an organism, it's going to be in a watery environment, and it will be in the ring form. So it's very, very rare to have like a linear sugar in water or in an organism, because organisms are full of water. So when you put that sugar in the water, it's going to react with itself to make a ring. And at the top of the screen here, I kind of have an example of that type of reaction. So here we have glucose, and this is the linear version of glucose. So when you have glucose as a powder, it would look like this. When you put that glucose into water, it's going to wrap around itself and make a ring. When it's doing that reaction to form the ring, you're always going to have the carbonyl carbon reacting with one of the other hydroxyl groups in the sugar. So always the carbonyl carbon and some hydroxyl group in the sugar. In this case for glucose, it's going to be the hydroxyl group that is on carbon 5. So when you put the glucose in the water, that line that it makes is going to kind of wrap around itself so that this hydroxyl group on the 5th carbon is kind of coming close to the carbonyl group. And you're going to have a reaction between this oxygen on the hydroxyl group and the carbonyl group carbon. So you're going to actually form a bond between this oxygen and this carbon. So in the products, you can see that bond having been formed. As part of the reaction, the hydrogen that was part of the hydroxyl group is going to end up transferring to the carbonyl group oxygen. So you can see that now you have a bond between the hydroxyl oxygen and the carbonyl carbon, and the hydrogen from the hydroxyl has transferred to the carbonyl oxygen, which means that you have a new hydroxyl group now, basically is what it means. Where you used to have a carbonyl group, now you have a hydroxyl group instead. So there is no more double bonded oxygen. Instead, you have an oxygen attached to a hydrogen, which is a hydroxyl group. So there's a couple of different ways that this reaction can go, or a couple of different types of ring sugars that you can end up with. You could end up with this one where the hydroxyl group is kind of pointing down, or you could end up with this one where the new hydroxyl group is pointing up. So anytime you have a linear sugar and you put it into water so it makes a ring, there's always two different rings that you could form. Always you could form one where the new hydroxyl group points up, and you could form one where it points down. And we have different names for those different rings. One of them is the alpha ring, and one of them is the beta ring. So we call it the alpha ring if the new hydroxyl group that you formed is pointing down. So on top here, where this new hydroxyl group is pointing down, this is the alpha ring form of glucose. So we call that alpha glucose. On the bottom, we have the new hydroxyl group pointing up. So that's going to be the beta form of the ring, and that's we're going to call that beta glucose. So the actual definition of the alpha ring versus the beta ring is actually more complicated than just whether the new hydroxyl group is pointing up or down. But for us, that's good enough because we don't need to get like it's already complicated enough probably. So we're not going to worry about the full definition of it, but just for our purposes, if that new hydroxyl group is pointed down, that's the alpha version. It would be alpha glucose or alpha fructose or alpha whatever. And if the new hydroxyl group is pointed up, then that's the beta version of the ring, beta glucose or beta fructose or beta what have you. So we've talked about the structure of monosaccharides now. For the other types of carbohydrates, for disaccharides, oligosaccharides, and polysaccharides, they all involve multiple monosaccharides that are linked together. And anytime monosaccharides are bound together, it's going to be using a special type of bond called a glycosidic linkage or a glycosidic bond. So just like two nucleotides would be joined together by a phosphodiester linkage or a phosphodiester bond, two monosaccharides are joined together by a glycosidic linkage or a glycosidic bond. In a glycosidic linkage, you have the hydroxyl groups, or two of the hydroxyl groups, on the different monosaccharides, so one on each monosaccharide, react with each other and release a water molecule, and they end up with just the sugars joined together by an oxygen atom. So that's shown in this figure here. On the top you have your two monosaccharides. These are actually identical. They're both alpha glucose. They're called alpha glucose because the new hydroxyl group that formed when this glucose molecule made a ring is pointing down in both cases. When these two glucoses react with each other, they're going to form a glycosidic linkage. You're going to have these two hydroxyl groups that are highlighted in blue reacting. So the entire hydroxyl group on one glucose will leave and join with the hydrogen from the other hydroxyl group to make a water, and then you're just left behind with that oxygen, which is going to end up attached to the carbon on the first glucose, like so that you have just these two monosaccharides attached to each other by a oxygen atom in the final glycosidic linkage. So in this example, you have the hydroxyl group on the first carbon of one alpha glucose reacting with the hydroxyl group on the fourth carbon of another alpha glucose. you can actually have different hydroxyl groups involved in the reaction. For different monosaccharides, different ones might be involved. And you're going to have different names of the linkages depending on which hydroxyl groups had reacted with each other. So they'll be named with numbers for the carbons that each of those hydroxyls was attached to. And you also have a symbol in the name, either an alpha or a beta, to let you know what is the orientation of the sugars in the bond. So in our example here, this is an alpha 1,4 glycosidic linkage between these two glucoses. We call it a 1,4 linkage because it's between the first carbon on one glucose and the fourth carbon on the other glucose. So those are the two hydroxyl groups that initially reacted to form the glycosidic linkage. And then the symbol, the alpha, indicates that both glucose molecules are facing the same way in the bond. So you can see each of them has their last carbon, their sixth carbon, kind of sticking up above the ring. And so for both of these glucoses, that last carbon is above the ring, not below the ring. So since they're both facing the same way, we call that the alpha glycosidic linkage. So the full name of it would be an alpha 1,4 glycosidic linkage, indicating that both of the monosaccharides involved are facing the same way, and that the bond is between the first carbon on one and the fourth carbon on the second. For any disaccharide, the structure of that disaccharide is going to depend on the type of glycosidic linkage that are linking its two monosaccharides, and it will depend on the identity of those monosaccharides. And it's the same thing for the other saccharides, the oligo and polysaccharides. Their structure is going to be determined by the types of glycosidic linkages that make them up, and also by the monosaccharides that are joined by those linkages. So the two most common glycosidic linkages that you can have are the alpha 1,4 glycosidic linkage, which we just saw on the previous slide, and the beta 1,4 glycosidic linkage. For both of these linkages, the carbons involved are the first carbon on one sugar and the fourth carbon on the other sugar. For the alpha glycosidic linkages, both of the sugars are facing the same way. Both of them are kind of facing the right way up. And that ends up with you having the oxygen in the glycosidic linkage pointing down. So the sugars are both facing right way up and the oxygen is pointing down. In the beta 1,4 glycosidic linkage, one of the sugars is facing up, but the second sugar is facing down. So they're facing opposite directions. And the oxygen in the glycosidic linkage is actually facing up, not facing down. So you're going to get an alpha glycosidic linkage anytime you have two alpha sugars, alpha monosaccharides, reacting with each other to form the linkage. You would get a beta glycosidic linkage anytime you get two beta monosaccharides reacting together to form the glycosidic linkage. But you can always recognize a alpha from a beta glycosidic linkage by looking at the monosaccharides and seeing if they're both facing the same way, then it's an alpha linkage. Also, if that oxygen is pointing down, then it's an alpha linkage. If the sugars are facing opposite directions, then it's a beta linkage. Also, if the oxygen is facing up, then it's a beta linkage. So now we'll go through several of the most important polysaccharides, and we'll talk about their structure and their function. And then at the end, we'll get kind of into the more general functions of carbohydrates. So the first we'll talk about is starch, which is commonly found in plants. Plants use starch to store energy, and it's basically alpha glucose monosaccharides that are linked together using alpha 1,4 glycosidic linkages to create this large polysaccharide. Whenever you have glucoses linked together using those alpha 1,4 glycosidic linkages, if you have a lot of them, it's actually going to create a structure that kind of coils around itself to form a helix. So starch is always kind of coiled up to make a helix. You can also have branches in starch. So some starches don't have branches, but other starches do. Anytime that you would have an alpha 1,6 glycosidic linkage linking two saccharides, instead of the alpha 1,4, there you're going to get a branch. And we have two basic types of starch depending on if they're branched or not. So amylose is starch that is not branched, so it has only the alpha-1,4 glycosidic linkages, and amylopectin is branched starch, which means that it has mostly the alpha-1,4 glycosidic linkages, but it also has some of the alpha-1,6 glycosidic linkages, which would be introducing branches. Glycogen is kind of similar to starch. It's also used for energy storage, but it's not found in plants. Instead, it's found in animals. For humans, the main place where you store glycogen in your body is the liver, but your muscles will also store it. And it's basically just a way for you to store glucose kind of in the long term and then access it when you need it. So similar to starch, glycogen is also made out of alpha glucoses with the alpha-1,4 glycosidic linkages, and it also has branches. Every time that you have a branch, that branch is created by an alpha-1,6 glycosidic linkage, so that's also just like starch. So the structure of it is really, really similar to starch. The difference, one of the main differences is that in glycogen, you actually have a protein at the very center of it. So this diagram here is basically illustrating a glycogen molecule, and the colorful thing in the center is actually a protein, and then the glycogen is kind of built out from that protein. So you have these chains of alpha glucoses bound together with the alpha-1,4 glycosidic linkages leading out from that central protein. And then of course you have a lot of branches. Everywhere that you have a branch, that lets you know that at that location you have an alpha-1,6 glycosidic linkage. So starch, even if it has branches, it usually doesn't have like that many branches. Glycogen has a bunch of branches though, so it has a lot more branches than starch does. That basically means that it's easier to kind of break glycogen down to get glucose out of it, so that if you need energy, you can get that energy very quickly. Basically, every place on this molecule where you have a chain ending, that's a place where the body would be able to break off a glucose molecule and use it for energy if need be. So the more of these branches that you have, the more chains you're going to get, and all of those chains will have an end. So the more branches you have, the more chains you have, the more places you have where you're able to break off a glucose molecule when you need it to be used. And then let's see, the picture at the top here is actually a microscope photograph or a micrograph of a liver section. So these kind of red cells are hepatocytes, which is just a liver cell. The blue balls in the middle are their nuclei. And actually the red stuff is glycogen. So these cells, I mean, they have a lot of functions, but one of their big functions is to create glycogen and store it, and they're just full of glycogen. Then anytime that you need energy, they're going to start releasing that glycogen, breaking it down and getting glucose back out of it, then releasing that glucose into your blood so you can get kind of a burst of energy. So this table is kind of showing you the difference between the structure of starch and glycogen. You have starch on the top here, glycogen on the bottom. Here you have amylose, which is unbranched starch. So it's just a single chain of alpha glucoses linked with the alpha-1,4 glycosidic linkages, and you can see that it twists around itself to make a helix. Anytime you have glucoses linked by the alpha-1,4 linkages, it's going to give you this coiling structure. Next to the amylose, you have amylopectin, which is branching starch. So it's also mostly just chains of alpha glucose joined by the alpha-1,4 glycosidic linkages, but you also have this one point where you have a branch. At that point, you have a glucose attached to another glucose using an alpha-1,6 glycosidic linkage. And if you were to zoom in on that point, you might see something like this. So here you have a couple of glucoses that are attached by the alpha-1,4 glycosidic linkage, and here you have glucoses that are attached by the alpha-1,6 glycosidic linkage. So this is a bond between the first carbon of one glucose and the sixth carbon of the next glucose, which means that it's that that carbon is kind of not part of the ring; it's sticking out above the ring. So whenever you make a bond using that carbon, you're going to end up with your new glucose that's attached to it kind of pretty far up above the first glucose. which gives you basically the branch, and you can then start a new chain off of this glucose. Then on the bottom, you have glycogen, which is structurally very similar to starch, also just alpha glucoses linked with the alpha-1,4 glycosidic linkages, and then whenever you have a branch, it's the alpha-1,6 glycosidic linkage that is forming that branch. But with the glycogen, you can see that there's just a bunch more branches than you have in starch. So a lot more branches means that you can get energy out of it a lot faster because you can break a glucose off from any of the ends of all of these different chains that it has. The next polysaccharide we'll talk about is cellulose, which is used for structure, not for energy storage. It is found in plants, and the main structure that it forms is the cell wall for plant cells. So animal cells do not have a cell wall, they just have a membrane around them and that's it. But plant cells all have a cell wall, which is outside of the cell membrane, and it's just a hard layer around the cell. It's kind of similar to the cell walls that we talked about for bacteria, which would be the gram-positive and gram-negative cell walls, but its structure is different. Its function is the same, but its structure is different. It's made out of cellulose. And cellulose is made out of beta glucoses that are linked together with the other super common type of glycosidic linkage, which is the beta-1,4 glycosidic linkage. So in starch and glycogen, you have alpha glucoses with alpha-1,4 glycosidic linkages. In cellulose, you have beta glucoses with beta-1,4 glycosidic linkages. That is kind of a small difference in terms of what it sounds like, but the implications of that difference are significant. The beta-1,4 glycosidic linkages that you find in cellulose cause you to form a straight chain instead of a coiled chain like you have in starch or glycogen. And it's also kind of difficult for a lot of creatures to break down the beta-1,4 glycosidic linkage. Basically almost any organism can break down an alpha-1,4 glycosidic linkage like you find in starch and glycogen, but not that many organisms can break down the beta-1,4 glycosidic linkage. Actually, there are no animals that can break down that bond at all. There are bacteria that can do it. So animals have no way to digest cellulose, basically. There are animals that kind of make their living by eating cellulose, like cows and other animals that graze and eat grass. Most of the nutrients in grass are just in cellulose, but they're not able to digest the cellulose themselves. Instead, they have bacteria living in their stomachs that will digest the cellulose for them. So no animal is able to break a beta-1,4 glycosidic linkage without the help of some other organism that would do it for it. So the figure here is kind of showing cellulose and how it is structured in plants to form the cell wall. At the bottom here, you have 4 different chains of cellulose that are shown. So these are the beta glucoses, and they're joined together with the beta-1,4 glycosidic linkages so that they're each one is facing the opposite direction from the glucoses that are right next to it. And that means you get straight chains, not helices. Because these chains are straight, they can line up with each other like parallel like this, and they can also hydrogen bond with the adjacent chains. And that lets you build very strong structures out of them. So when plants are using cellulose to build their cell wall, they're going to get a bunch of chains of cellulose running the same way as each other, running parallel. They're all going to be hydrogen bonded with the chains that are next to them, and that kind of is going to form a microfibril. Then the plant will get a bunch of microfibrils together to make a macrofibril, and get a bunch of macrofibrils together to make an actual cellulose fiber. Then if you use a really fancy microscope to take a picture of the cell wall of a plant, you can actually see those cellulose fibers running across it. It's basically just composed of a bunch of these cellulose fibers just woven together like this. So here you have a diagram of that structure of cellulose. These are the beta glucoses in cellulose. Here you have 2 different chains that are shown. So you have one chain on top here and a second chain on the bottom. These are the beta-1,4 glycosidic linkages. So you can see that carbon 6 points up on the first glucose in each of these chains, and that means it's going to point down on the second glucose. Since it's a beta 1,4 glycosidic linkage, each of these glucoses needs to be facing the opposite direction. Because you have some of these 6 carbons facing down, that lets you do hydrogen bonding between adjacent chains. So you actually have hydrogen bonds that form between the hydroxyl on one of these carbons on the first chain and the hydroxyl on the 6th carbon on the second chain. And it'll kind of alternate like that. So you're always going to have the 6th carbon's hydroxyl involved in a hydrogen bond with a chain that's above or below it. Then if you zoom out, you see a structure kind of like this. So here you have multiple chains of cellulose. They are all straight because of the beta 1,4 glycosidic linkages that the glucoses are bound together using. And in the kind of dotted yellow lines, you can see there's hydrogen bonds occurring between each of these sugars in the chains. So each one of these chains has many hydrogen bonds that it's forming with the chains that are above and below it, and that makes a very, very strong structure. The next polysaccharide we'll talk about is chitin, which is similar to cellulose in its structure and its function. So it's also used to form cell walls, but not by plants. Instead, it's used for the cell walls of fungi, which would be like mushrooms. It's also used to make the exoskeleton for insects and crustaceans. So here you have these mushrooms. The cell walls for each of these mushrooms would be formed by chitin, and for this insect, the exoskeleton it's leaving behind, as well as the new one that it's made, is all made out of chitin. So similar to cellulose, chitin is very, very strong. It's also joined by sugars that, well, this is also formed by sugars that are joined by the beta 1,4 glycosidic linkages. Those linkages will give you a straight chain, and they're also pretty hard for a lot of organisms to degrade. So that makes it strong. The difference between chitin and cellulose is that cellulose is made out of beta glucoses and chitin is made out of a different kind of sugar called N-acetylglucosamide, or NAG. So N-acetylglucosamide is actually just a glucose molecule that is kind of modified. It has an amine group on it and an acetyl group on it. You don't really need to worry about that. We'll just call it NAG. It's just a different type of monosaccharide. But you're getting all these NAG monosaccharides, linking them together with the beta 1,4 glycosidic linkages, which gives you a straight chain. And then because a chain is straight, you can kind of stack it on top of other chains, running parallel with those other chains, and you can have hydrogen bonding between the chains that are next to each other, and that again gives you a very, very strong structure, just like cellulose. So here is a diagram showing you kind of that structure. These are the N-acetylglucosamide sugars that are the monosaccharides for chitin. So they're just abbreviated NAG. These are both beta NAGs, which means that when they make a glycosidic linkage together, it's going to be the beta glycosidic linkage. So they're forming beta 1,4 glycosidic linkages together, and you can see that they're each facing a different direction. So in blue here you can see kind of the extra group that's been added to the glucose to make it an NAG monosaccharide. This is just an amine group with an acetyl group attached to it. And you can see that of course they're pointing opposite directions because this is the beta glycosidic linkage. Over here in the zoomed out structure you can see multiple chains of chitin lined up with each other. Each one is formed by the beta NAGs linked together with the beta 1,4 glycosidic linkages, and you can see the little blue modification to the sugar sticking off in opposite directions for each alternating sugar. And you can also see the hydrogen bonds in yellow, or the dotted yellow lines between the chains are hydrogen bonds between different sugars in the chains. So similar to cellulose, you have straight chains that are lined up parallel to each other and attached to each other using hydrogen bonds. Because you have a lot of different hydrogen bonds attaching the chains, that makes it a very, very strong structure. So the last polysaccharide that we'll talk about is peptidoglycan, which we've already mentioned in connection with bacterial cell walls. So peptidoglycan is really similar to cellulose and to chitin in terms of its structure and function. It's also used to build cell walls, just in bacteria instead of in fungi or in plants. It's also consisting of straight chains of the polysaccharide that are linked together, and each chain is composed of monosaccharides that are linked together using the beta-1,4 glycosidic linkages. The difference that you have between peptidoglycan and cellulose or chitin is going to be the monosaccharides that it's composed of, and also the way that different chains are attached to each other. So in cellulose, you have beta-glucoses making up each chain. In chitin, you have beta-NAGs. In peptidoglycan, you have the beta-NAGs, and you also have another type of monosaccharide, which is N-acetylmuramic acid, or NAM, also the beta form of that. So NAG is just a modified glucose. N-acetylmuramic acid is also another modified glucose. It actually has, even another group added to it on top of what the NAG has added to its glucose. So that forms NAM. So they're both basically just modified glucoses. So you'll have an NAG, then an NAM, then an NAG, then an NAM. They just alternate. They're always attached to each other using the beta-1,4 glycosidic linkages. And whenever you're building a polysaccharide using that type of bond, you always get a straight chain. Each NAM monosaccharide or each NAM sugar will also have a really short chain of amino acids attached to it, just coming off of the ring, so it has a little peptide on it. And then instead of the chains being attached with hydrogen bonds the way that they are in cellulose or in chitin, in peptidoglycan you actually have those little bits of peptide hanging off each NAM sugar attaching to each other. Those are going to be attached using actual covalent bonds, which makes peptidoglycan extremely strong. So here's your diagram for peptidoglycan. It's just on the alternating pattern of these two sugars. So you have your beta NAM, then your beta NAG. The NAM sugar also has a little peptide attached to it, which is shown in green here. So each of these green blocks is an amino acid, and it has just kind of this 4 amino acid peptide hanging off of it. And then these sugars are always attached using the beta-1,4 glycosidic linkage, which means that they're going to be facing all opposite ways each time. If you zoom out on that structure, you're going to see your straight chains of peptidoglycan. Anytime you build a polysaccharide with the beta-1,4 glycosidic linkages, you get a straight chain like this, in which each sugar is facing the opposite direction as the sugar to either side of it. These chains are going to be stacked on top of each other, and they're not going to be attached with hydrogen bonds. Instead, they're going to be attached with covalent bonds between the peptide bits. So you can see in green the little peptide bits hanging off of each NAM sugar. Some of those are going to end up overlapping each other on adjacent chains of peptidoglycan, and you can get covalent bonds forming between those little bits of peptide. So because these chains are actually attached to each other using covalent bonds, this is an extremely strong structure that's formed by the peptidoglycan. So now we'll just kind of go over some more general functions of carbohydrates, rather than referencing specific carbohydrates as much. So the general categories that these functions fall into are forming building blocks in the cell, providing structural support for cells or organisms, helping cells kind of signal their identity or display their identity to other cells and communicate with each other, and also, of course, storing energy, which is the main function of carbs. So, of course, all nucleotides contain a sugar, either RNA for, either ribose for RNA or deoxyribose for DNA. Here you have an example of a nucleotide, and the sugar in it is shown right here. So this is going to be deoxyribose because it doesn't have an OH. It doesn't have an oxygen on its second carbon. So of course that means you need sugars so you can make nucleotides. But you also have other molecules in the cell that are really similar to nucleotides that are basically derived from nucleotides. For instance, you have ATP, which is shown right here. So you can see that that's really, really similar to a nucleotide. Actually, the main difference between ATP and a nucleotide or an adenosine nucleotide is just the fact that in ATP you have 3 phosphate groups on it, not just one. So you can have a ribose sugar in that adenine, in that ATP as well, which means that you need sugar to build it. Then also your electron carriers, a lot of them look really similar to nucleotides, which means they also have sugars in their structure. So you're going to have sugars in order to build all of those things that are required by the cell, ATP and electron carriers and nucleotides for your DNA and your RNA. So if you don't have those sugars, you're not making any of those things, and the cell is going to die for sure. But you can also make other things with sugars. You can actually kind of strip a sugar down, get the hydroxyl groups off of it, and just leave yourself with a chain of carbons. Then you can use that chain to build other molecules that have a chain of carbons, which is a lot of them. So for instance, you can build amino acids from sugars. And the cell will do that if it needs to, to make more amino acids and have sugar readily available. Another role of carbohydrates is to provide structural support. Usually it would be polysaccharides that are involved in providing structural support. We already went through kind of the 3 main ones, which are chitin, cellulose, and peptidoglycan. For any of these polysaccharides that is functioning in structural support, it's going to have certain features. It's not going to have any branching in the chain of the polysaccharide. It'll just be straight molecules, and you're going to have the monosaccharides that are making up that polysaccharide linked together using the beta glycosidic linkages. It won't necessarily be beta 1,4 for every single structural polysaccharide. It's beta 1,4 glycosidic linkages for chitin, cellulose, and peptidoglycan, but there's also other structural polysaccharides out there. But it's always going to be some type of beta linkage, because when you're using the beta glycosidic linkage, that gives you a straight chain, and it's also hard to break that linkage. There's just not that many organisms that have the enzyme you need to break that bond down. No animals can do it, for instance. So you're going to have, just always a straight chain, no branching, the monosaccharides linked by the beta glycosidic linkages, and then you're going to have like these chains being stacked on top of each other, and they'll have some type of interaction between the adjacent chains. Maybe it's a hydrogen bond, maybe it's some other type of bond, but something to hold those adjacent chains together. So all structural polysaccharides are going to have those features, which just kind of help make them strong. Carbohydrates are also used to establish cellular identity, and that's always occurring in carbohydrates that are attached to the membrane somehow. So we haven't talked about the cell membrane yet in any detail. We'll go into it in the next chapter, but the cell membrane is composed of lipids, and it also has a bunch of proteins embedded in it. So a lot of those lipids and proteins are going to have oligosaccharides attached to them. If you have a lipid with an oligosaccharide attached, we call it a glycolipid. If you have a protein with an oligosaccharide attached, we call it a glycoprotein. The glyco there is meaning sweet or meaning sugar. So anytime you see a word that has glyco in it, anywhere in it, you know you're looking at something that has a bit of sugar in it somewhere. So here you have kind of a diagram of a cell membrane. These blue balls with the little legs coming off of them, these are the lipids that are making that membrane up, and in green you have proteins that are embedded in that membrane. And a lot of these lipids and proteins have, in red, oligosaccharides coming off of them. These oligosaccharides are going to be used to kind of show off or display the identity of the cell to other cells. All of these oligosaccharides are going to be different, or there's many different types that would be used. So you can see that they actually have different little patterns of branching in them, but not just that, the actual monosaccharides that make them up are going to be different, and they're kind of arranged in different patterns. So you have a lot of different monosaccharides that are used to build these oligosaccharides arranged in different patterns, and you have different patterns of branching. So you can really make a lot of unique oligosaccharides. Since you can make a lot of them that are unique, it's pretty easy to use them as kind of a marker of cell identity. So different cell types are going to have different oligosaccharides on them. For all of your cells, there's going to be certain oligosaccharides on those cells that kind of mark them as belonging to you. And then there's also going to be other oligosaccharides that are just marking like what type of cell it is. So if you're looking at like a red blood cell versus a white blood cell, they're going to share some of their surface oligosaccharides that are just saying that, this cell is belonging to you. But there's also going to be some oligosaccharides that are different between them that are signaling that the red blood cell is a red blood cell, whereas the white blood cell is a white blood cell. So every cell is going to have a bunch of those oligosaccharides on its surface that are specific to the cell type that it is and the organism that it's a part of. You also have some oligosaccharides on your cells that mark you as human. So different species will have kind of certain different oligosaccharides that are identifying the cells in their bodies as belonging to that species. Then they'll have other oligosaccharides that identify the cells belonging to a specific individual. And again, as I mentioned, even other oligosaccharides that mark what cell type each individual cell belongs to. That ends up being really important for communication between cells, also for the functioning of your immune system. So your immune system is actually going to be scanning those oligosaccharides that are on the surface of cells to tell the difference between a cell that belongs to you and a cell that does not belong to you. So anytime your immune system finds a cell that has oligosaccharides that it's not familiar with, it's going to figure that cell is not yours and is going to attack it as a foreign invader. It's also useful for cell communication. Sometimes one cell needs to send a message to other cells that are of a specific type, and it can target that message to the specific cell type it wants to send it to using the oligosaccharides that are on the surface of those specific cells. Finally, this kind of oligosaccharide cell identity system is important for cells recognizing each other during mating. So when you have mating and sexual reproduction, where a sperm comes to an egg and it's going to try to kind of penetrate that egg and fertilize it, first the sperm needs to be able to actually attach to the egg. And the way the sperm is going to attach to the egg is by having a receptor on its surface that is able to bind to specific oligosaccharides on the surface of the egg. The oligosaccharides that the sperm is able to bind to are going to be for the specific species that the sperm is from. So if it's a human sperm, it's going to be looking for human oligosaccharides on the surface of the egg. If it finds a different oligosaccharide there, it's not going to be able to attach to the egg. So this prevents species from kind of reproducing with each other, between species. This prevents interspecies reproduction, basically, because sperm and eggs of different species don't have the right oligosaccharides to recognize each other. I'm sure you all probably know about the importance of blood type in determining whether it's safe for you to get a particular or to get a transfusion from a particular person. And it's good to know your blood type. That blood type system is all based on oligosaccharides on the surface of your red blood cells. So everybody is going to have either type A blood, type B blood, type AB blood, or type O blood. That A, B, and O, those are all different oligosaccharides on the surface of your red blood cells. So that's kind of shown in this diagram here. Everybody starts off life with red blood cells that have the O oligosaccharide on their surface. And the O oligosaccharide is shown right here. It's this little diamond-like pattern of 4 different monosaccharides bound together. And you can see the identity of those monosaccharides down at the bottom. Then, depending on what genetics you have, you may or may not have specific enzymes that are able to modify this oligosaccharide and add something extra to it. You might add a galactose sugar to it right here, which would give you the B oligosaccharide, or you might have enzymes that add an NAG sugar to it, which would give you the A oligosaccharide, or you might have both enzymes, and then you're going to end up with AB blood. So you'll have some of the A oligosaccharides and some of the B oligosaccharides. Or you might not have either enzyme, and then you just stay with the type O blood. So the immune system is able to recognize the A oligosaccharide and the B oligosaccharide. It can't recognize the O oligosaccharide because it's just kind of too simple. Its structure is just a little bit too simple for the immune system to specifically recognize it. But if you have type B blood, then your immune system is going to learn that the B oligosaccharide belongs to you. It's going to learn that, that's the oligosaccharide that is found on the surface of my red blood cells. So then if you got a transfusion from a person who has type A blood, then your immune system is going to notice the new oligosaccharide on that new blood cells. Or on the cells of that new blood. It's going to notice that A oligosaccharide, and it's going to know that it doesn't belong to you, and it's going to attack those new cells. That's going to mean that you're not getting the benefit of that transfusion. All those new cells, all those new red blood cells that you got, they're kind of dying as your immune system kills them, but it actually might be even worse than that, because if the immune response gets like kind of too intense, it can actually cause inflammation all over your whole body, and that could actually drop your blood pressure and kill you. So that's kind of the reason why it's really important to make sure you're getting transfusions from a person with a compatible blood type with you. And it just means, a person whose blood your immune system isn't going to recognize as being foreign and then attack. So if you have type O blood, that means you can actually donate blood to anybody, but you can only receive type O blood. Your immune system will not have learned the B oligosaccharide or the A oligosaccharide. So if you get type A or type B blood, your immune system is going to identify that new oligosaccharide and attack it. If you get type O blood, that's kind of the only way to get blood that doesn't have oligosaccharides your immune system would respond to. If you had type AB blood, on the other hand, you would only be able to donate blood to someone else who has type AB blood, but you could actually receive blood from anybody because your immune system will already be familiar with the A oligosaccharide and with the B oligosaccharide, since those are both normally found on the surface of your own cells, and it'll recognize them as belonging to you. So then let's say you got a transfusion from a person with type B blood. The new cells will just have the B oligosaccharide on them. The immune system will come and find that B oligosaccharide, and it'll say, everything's good here, that's one of mine, and it won't attack. So, so you'll be good. So the last function of carbohydrates we'll talk about is their main function, which is energy storage. And so first we'll just try to understand why it is that carbohydrates are able to store a large amount of energy. And it's basically because they have a lot of nonpolar bonds. Those nonpolar bonds tend to be very long. In general, when you have a bond that is longer, it has more energy associated with it, and it's also going to be weaker. A bond that is shorter is going to have less energy associated with it and be stronger. So a nonpolar bond is always going to be longer than a polar bond, or I should say almost always. A nonpolar bond is almost always going to be longer than a polar bond. Since it's longer, it has more energy and should be weaker actually. But in water, a nonpolar bond is actually not weaker than a polar bond because you're going to have water molecules kind of tugging on the atoms involved in a polar bond. Since both those atoms are going to have a partial charge on there, there's just kind of a place for water to kind of get a grip and give a little tug, and that's going to weaken the bond in water. So in water, nonpolar bonds are stronger even though they're longer than the polar bonds, but they are still more energetic because they're longer. So in a typical carbohydrate, you have a bunch of those nonpolar bonds between the carbons and the hydrogens and between the carbons and the other carbons in the molecule. Those are all nonpolar bonds. Those are all bonds that are storing a large amount of energy because they're so long. If you look at carbon dioxide, on the other hand, it's nothing but polar bonds. Those bonds are going to be shorter, and they're going to be less energetic. So of course, your body isn't able to actually use the energy that is stored in carbohydrates directly to power chemical reactions. It actually has to take that energy and convert it to ATP, or use it to build ATP, and then it can use ATP as a source of energy to power basically any chemical reaction that the cell needs to get done. So later in the semester, we're going to go over the process of how monosaccharides, or specifically glucose, is used to create ATP. It's a really long process, but you can kind of summarize it with this brief chemical equation in which you have glucose and you combine it with oxygen, and that gives you carbon dioxide, water, and energy. Except in this case, this energy would be not in the form of fire, but it would be in the form of ATP. You would be using that energy to attach the last phosphate group to an ADP molecule, which gives you a TP. In this process, you're starting out with, on the reactant side, a glucose that has a lot of long nonpolar bonds. Since those bonds are nonpolar, that means they're long. Since they're long, that means they have a lot of energy. At the end of the process, these carbons that started off in glucose are going to end up as carbon dioxide. The hydrogens that started off in glucose are going to end up in water. So in both cases, you have atoms that used to be in nonpolar bonds in glucose ending up in polar bonds in some other molecule, either carbon dioxide or water. So they're ending up in these polar bonds that are shorter. Because the bonds are shorter, they have less energy. So you're starting with high energy bonds in glucose, and you're going to low energy bonds in carbon dioxide and in water. That extra energy that is lost is shown in this picture as a little fire, but it's actually being used in your cells to attach a free phosphate group to ADP, which gives you ATP that can be used to power any other reaction. So when your body needs to use carbohydrates to make ATP, it's always going to use monosaccharides to do that. It'll be glucose, maybe a different monosaccharide, but always a monosaccharide. If you have a larger carbohydrate, if your body's going to use that for energy, it needs to chop it up into the component monosaccharides. Then it can use those to make ATP. But if your body is going to try to store some of that energy for later, it will not do it using monosaccharides, just kind of like free glucose floating around. It'll instead build some type of polysaccharide. So if you're a person, your body's going to be building glycogen. But if you were a plant, then your body would be building starch instead. So those are the two polysaccharides we went over that are used for energy storage. In both cases, those polysaccharides are built out of alpha glucoses that are joined together with the alpha-1,4 glycosidic linkages. They're basically used to keep glucose kind of safe and sequestered for long-term storage. Then later on, if you have kind of a glucose shortage and you need to make more ATP, you can just go to your starch or your glycogen and start chopping it up, get the glucoses out of it, and then you can use them to make ATP. So wherever you have a chain ending in one of these polysaccharides, that's where you're going to be able to have an enzyme come and break a glucose off the end of the chain, and then you can use that glucose, to build ATP. And you have a couple different enzymes that would be responsible for that. In your own body, you have the enzyme phosphorylase, which is responsible for breaking the glycosidic linkages in glycogen. It's always going to break at the ends. So for the last glucose that's in a chain, phosphorylase is able to break the glycosidic linkage between that glucose and the next glucose in the chain, which means that the more ends to the chain you have, the faster that enzyme is going to be able to mobilize the glucose and glycogen for use. In starch, in plants that have starch, the enzyme they use is amylase.